So today we're going to talk about components of the lymphatic system. So we're pretty much done for the time being, at least, in talking about the basic types of histological tissues. So we've covered muscle, we've covered connective tissues, we've covered epithelia. The last couple of lectures in this course, we will cover the nervous system and the nervous tissues. And so today we're going to talk a little bit about how some of the things that we've talked about so far can be put together to make a larger structure, like an organ that we're going to be looking at today. But before we do, seeing this picture before in your notes, and that was at the end of the lecture on blood vessels. Unfortunately, I didn't get a chance to go over it. But I did promise that I would get back to this. And today is when this is becoming relevant. Because we've covered blood vessels, we've covered blood, we've covered inflammation. And so, what happens when you have inflammation? Well, one of the first things that happens during an inflammatory response is swelling. You're going to have a lot of liquid leaving the bloodstream into the connective tissue. How is that liquid, how is that fluid going to be taken back in? Is the question. And that's where lymphatic vessels become important. Let me just check to see if we have a drawing of this somewhere. We'll just redo it. Probably easier that way. So what I'm going to do is I'm just going to try to reproduce this image here, so feel free to draw along with me if you'd like, or you can just scribble over the image you already have. I just want to be able to take some of what's on there and talk a little bit about it. So again, we're going to have some sort of an artery coming along, branching off into an arteriole. That arteriole can act as a precapillary sphincter, and then open up into a network of capillaries. So we've got a whole bunch of capillaries here, all of them branching off from an artery, and then we have it draining into initially post-capillary venules, then which become larger venules, which will then merge together into drain into a vein. So basically that's part of what you already have up there. This is what you've talked about so far. We have vein, venule, post-capillary venule, artery, arteriole, and capillary. So let's talk a little bit of physiology. Actually, interestingly enough, a few days ago I had a student drop by my office, a former student drop by my office. He's in medical school right now. And he dropped by my office just to say hello and to tell me that he learned a lot of physiology in my course. And I thought, I suppose a histology course, how did you learn a lot of physiology? Well, it seems I tend to include a bit of physiology in here, so I guess you'll learn a little bit of physiology with me as well. So, here's some physiology. When the blood comes into this system here, into this capillary network, it is at a fairly low pressure, but the pressure is still relatively high when we think about what this capillary can withstand. Okay, so what we have here is what is known as fairly high hydrostatic pressure. It's the one it is, okay. Which means that there's basically a lot of liquid, a lot of fluid passing into these vessels. Now, as you might imagine, if you try to push liquids at a fairly high rate through at a fairly high pressure, through a relatively thin walled vessel, there's going to be leakage. And so a lot of that liquid that's in there, a lot of this plasma is going to exit the bloodstream. Basically going to flow out into the surrounding tissues. Because it's relatively high pressure, and again, the capillaries can't really withstand a lot of that pressure that's coming in. And so a lot of the fluid will exit the blood vessels anyway, regardless of whether there is any kind of inflammation there or not. There's going to be some spillage. And that's normal. Now, as the liquid is leaving the interior of the blood vessel, what's going to happen to the contents of the blood vessel? It's going to get more and more concentrated. Okay? So which means that by the time the blood reaches this end here, we have high osmotic pressure. And low hydrostatic pressure. So there's very little liquid pushing against the walls of the blood vessel from the inside, and there is a lot of material inside those blood vessels that will tend to attract water back into the blood vessel. And so what we have here is a return of that fluid, that water that was lost initially, back into the venules. So again, these are just referring to what's going on inside of the blood vessel, not on the outside of the blood vessel. Okay? So again, initially at the beginning of the capillary system, you have high hydrostatic pressure, low osmotic pressure, and so liquid, especially water, will leave the blood vessels and into the connective tissues. Okay? As the blood is flowing through the capillary system towards the end, what you have inside the capillary is fairly high concentration of material, fairly low amount of liquid, and so it tends to draw the water back in. In fact, it will take about 90% of the fluid back in to the blood vessel. But there will be about 10% of the liquid that has left the capillary system. It will remain within the tissues. Yeah.
So can you say explaining how the water comes out?
Okay, so again, at the beginning, the initial portion in the capillary bed, you have high hydrostatic pressure, which means the blood is at a relatively high pressure. And so it presses against the walls of the capillaries. Now, your blood capillary walls are nothing more than just the endothelium. Those endothelial cells are just joined together by some occluding junctions and some adherence junctions just to hold them together. That's about it. Okay, so there's not a lot of strength there. And so there's going to be some leakage out of the blood vessel. Okay? So we have relatively low osmotic pressure, so there's really no influx of water into the blood vessel at this point because the contents are relatively low concentration. But as water leaves the blood vessel, the contents of the blood vessel become more and more concentrated. And so by the time you get to this part, the end part of the actual capillary bed, what you have is fairly high osmotic pressure inside the blood vessel and low hydrostatic pressure. So there's very little liquid inside it that's pressing against the walls trying to leave. In fact, a lot of the liquid from the tissues on the outside is trying to get back in to dilute the concentration of material inside the blood vessel. Okay? And so again, about 90% of the liquid that leaves out here is going to return to the blood vessel in this region here. So at the end of the capillary bed, 90% of the liquid returns back into the vascular system, or the blood vascular system. That leaves about 10% in the tissues that's still left over. Now, if you don't have a way of draining it, that 10% will just keep on building up and up and up, and you will have more and more liquid building up inside of those tissues. If you want to know what it looks like to have a blockage in the drainage system, look on Google for pictures of elephantiasis. Elephantitis. Go ahead and Google it right now if you've got a laptop in front of you, and you will see a picture, or many pictures, of people with legs that are just this thick. Okay? Because they're just engorged with fluid that just isn't draining back from the tissues. For those of you who are looking at it or making faces right now. So, yeah, that's what happens when you don't drain the fluid. So, what your body has is a drainage system. So whatever is left in that tissue, that extra 10% of fluid, is going to be able to enter into this capillary system of lymphatic vessels. These are going to be lymphatic capillaries. These are blind-ended vessels. So they just basically are distributed throughout the tissues, and they collect the fluid. So this is the lymphatic vessel. Now, if you look at your notes from the lecture on blood vessels, the last few deal with lymphatic vessels. One of the things about lymphatic vessels is that they are much more porous. They tend to have a fairly discontinuous basement membrane. The endothelium isn't quite as tightly packed or bound together as a typical blood vessel. And so they very much allow very easy re-entry of fluids back into that vascular system. So they allow the drainage of that excess liquid to enter into, instead of going into the blood vessels, the fluid can also enter into the lymphatic vessels. And so the remaining 10% can drain out through the lymphatic vessel system. And then eventually those lymphatic vessels become large lymphatic vessels, which will then end up just draining into the thoracic vein before it enters back into the circulatory system, before it enters into the heart again. Okay? So you do have this connected back to the venous system. And so you can have that fluid come back. Now, why is all of this relevant to today's lecture? Well, what if you have a pathogen here? So let's draw a pathogen, let's give you some things. So what if you have a pathogen here? All of a sudden, you have an inflammatory response occurring here. What do we have then? What's going to happen when you have inflammation? Sorry? Swelling. Good. So we're going to have an influx of blood. We're going to have swelling. So a lot more liquid will leave this enter the tissue. Because there's a lot more blood being brought into the area. Okay. What else is going to happen after that? What's the next thing? You're bringing in a lot of fluid. What else are you bringing? Inflammatory response. Cells. Thank you. Which ones are the first ones there? Neutrophils. Great. See, this stuff is starting to connect. It's starting to grow. Okay. So neutrophils will be the first ones coming into the area. They're going to margin. They're going to roll along the endothelium. They're going to transmigrate across that endothelium and use chemotaxis to find this guy right here and take him out. Okay. They're going to break him down into little bits, little fragments. Anything inside that cell, the proteins inside that cell, will be destroyed and basically dumped into that extracellular matrix because, again, neutrophils aren't exactly going to take it up and sequester it. They die after they're done doing their job, so they just chew it up, spit it out, and they die on the spot. So what you have left is basically a battlefield left with a lot of debris, a lot of fragments of cells and proteins. Some of them pure self proteins, and some of them are foreign proteins. Okay, so we have a lot of fluid, we have a lot of broken down proteins. What's going to happen is all that stuff will enter into the lymphatic system because that system is easily accessible. And again, the lymphatic system is there to drain the fluid away that is in excess. Okay? So there's going to be some water that will be taken back up by your blood vascular system. But again, a lot of that fluid is built up there to such a degree that you will need some help. And so your lymphatic system will take up all that fluid. And again, those fragments of proteins, because they are too big to enter the blood vascular system, they won't enter that, but they will be able to enter the lymphatic system. And where is this leading? Well, the lymphatic vessels eventually lead out towards the venous system, but before they get there, they go through a couple of lymph nodes, one or two maybe, before they enter the venous system again. Okay? So, and those lymph nodes are going to be there to clean it up. Okay? So, this is where the lymphatic system comes in. It's going to take this, all this stuff that's coming in from that inflammatory response, figure out what is foreign, what isn't, and then mount a response to the foreign particles. So going to notice that something here does not belong. We need to make sure that we have some sort of response ready for when we see this thing again. Okay, so let's get to our slides. So again, today's focus will be mostly on organs and lymphatic organs. So when we talk about lymphatic organs, we can subdivide them initially into primary lymphatic organs, and secondary lymphatic organs. Primary lymphatic organs are basically responsible for producing lymphocytes. Okay? So development of lymphocytes and their education, they're not really involved in dealing with infection. Okay? So they're not really involved in dealing with what we just talked about. They're just there to produce the lymphocytes, B or T lymphocytes. And so the primary lymphatic organs are the bone marrow, which is where B lymphocytes come from, and the thymus, where the T lymphocytes come from. Now, depending on who you ask or which sources you go to, you might also see the GALT, something named GALT, shown in there as well, which stands for gut-associated lymphatic tissue. Okay? So there will be some education of lymphocytes happening there as well. Okay? But again, that might also be involved with an actual response to a pathogen. Now, secondary lymphatic organs are the ones that are responsible in actually responding to the pathogen. So once you've found something that's foreign, that does not belong, your secondary lymphatic organs are the ones that will actually mount a response. So here we're talking about lymphatic nodules, which are the smallest things and not really classified as organs, but tissues. Then lymph nodes, which I just mentioned, tonsils, and the spleen. So in today's lecture, we'll talk about the thymus, we'll talk about the lymph node and the nodules, and we'll talk about the spleen. We won't really cover the others. Okay? So feel free to read about those, but you're not responsible for those tests. Now, before we can really get into a discussion of the lymphatic system, let's have a little crash immunology. Well, today's really going to be a crash immunology course, really, to some extent. So let's talk a little bit about antigen-presenting cells. Now, basically, you can think of any cell that presents antigens on the surface as an antigen-presenting cell. But what is an antigen? You have self-antigens, you have non-self-antigens. So technically, an antigen-presenting cell is one that is classified as a cell that has something called an MHC2 complex on its surface. Let's go to the drawing. So So let's talk about two cells. We'll make them egg-shaped just for fun. So they're just regular old cells. No one really knows what kind of cell it is. There's the nucleus. And what's going to be important about these cells is what we find on the surface. Now, just put these little brown molecules on their surface. I'm going to call them MHC complexes. Okay, so, I'm actually going to draw a couple more complexes on this one, just to make things more complicated, because things are often more complicated than I present them as. I'll tell you that it gets more complicated than I want to explain to you, but we're going to try to keep it relatively simple. So you've got two cells, apparently very similar to one another, but somewhat different because they have different cell surface molecules in some way. Okay, so these characters that I've drawn in brown are MHC1 complexes. So I put my M's. They stand for Major Histocompatibility Complex 1 and Major Histocompatibility Complex 2. It's a bit of a mouthful, which is why you need to stick with MHC. If you're looking at literature, if you're looking at journal articles, you will quite often come across something called LHA. And that's basically what it's referring to as well. The ones that I've drawn in black, represents the are MHC2. So, now we just defined antigen-presenting cells as cells that have MHC2 complexes on their surface. So this cell here would be an APC or an antigen-presenting cell. This cell, on the other hand, is pretty much any cell in your body, or any other cell in your body. Okay, so all cells will have an MHC complex on their surface, but which type will depend on what that complex is being used for. So, why do we have MHC1 complexes? MHC1 complexes present antigens synthesized inside the cell. MHC2 complexes present antigens of things that were actually taken up, engulfed by the cell, or phagocytized by the cell, and broken down. Okay, so this cell here will take up a pathogen or something from the outside. So let's draw our little guy with fangs. This little critter is going to get chewed away by hydrolytic enzymes. And so we're going to end up with a little vesicle containing fragments, bits and pieces of that pathogen's proteins. Okay? And it is those fragments that will then be presented on the surface of an MHC2 complex. Okay? So MHC2 molecules will present antigens that were external to the cell. So the cell had to take them up from the outside, not outside world, but outside within the tissue, from outside of itself, broken it up, and then presented it on the surface with an MHC2 complex. Okay? Whereas what cells normally do is they will produce a bunch of proteins. And most of those proteins are going to be there to perform a particular function within the cell. But some of those proteins will also be broken down by the cell into little fragments. Again. And those fragments will be presented on the surface with an MHC1 molecule. So your typical cell producing whatever protein it has, and of course we made that a different color just so it sticks out a bit more. Let's go with green. Okay, so this little protein is now green, has little green dots, and so our little green protein produces green little fragments that can be presented within MHC1 molecules on the cell surface. Okay. So again, any protein that is synthesized within this cell, or this cell for that matter, okay, there's going to be a small subset of that protein that will simply be naturally taken away, broken down into little fragments, and presented on the cell surface within an MHC1 molecule. And then an APC will also take foreign matter from the outside, break that down, and then present fragments of that on its surface within MHC2 molecules. Okay. So now think about it. If this is an APC, like for example a macrophage, it's going to chew away a bacteria, and take some of those bacterial proteins, some of the cell surface proteins found on the surface of that bacteria, and present them on the surface. It's going to present them within an MHC2 molecule, and it's going to be presenting that antigen to the lymphocytes that might be coming along. The lymphocytes will come along, they will attach to the MHC2 molecule, and they will scan whatever is within that MHC2 molecule to see, is this in fact foreign or not? And if it's foreign, they will be able to elicit some sort of a response. They will be able to become activated, potentially, so they can now find this target elsewhere and recognize it. Now, MHC1 molecules will show self-antigens, at least most of the time. I'm saying most of the time, will be self-antigens. Because, again, these are proteins produced within the cell. And so, again, some of those proteins will be shown on the surface to say, here's what I'm producing. And again, a lymphocyte might be coming along, and they will attach to that MHC1 molecule and check to see the contents to see, is it self or non-self? Under what conditions would it be non-self? Can you think of any time when an MHC1 molecule will be presenting a non-self molecule? Yeah?
A cell like a virus?
Thank you, a virus. Okay. So this is why I haven't said their own proteins. Proteins produced inside the cell. If it's a viral protein, it's going to be produced inside the cell. Some of those viral proteins, just like any other proteins produced by that cell, will naturally be degraded and produced and shown within an MHC1 molecule. And so some of these might actually be viral proteins. And again, your lymphocytes can come along and say, oh, this doesn't belong here. This cell is infected. Let's kill this cell. Okay. Or do something else. Okay. So we have now a way of presenting antigens by infected cells. Interestingly enough, some viruses have adapted to this by decreasing the number of MHC1 complexes on the surface of a cell that has infected. And in turn, our lymphocytes have adapted to this by learning to count MHC1 molecules on the surface. So they can actually detect how many MHC1 molecules there are on the surface, and if there is an abnormally low number, they know something's going on. And remember, all of this is done by cells that don't have eyes, don't have brains. They're just simply responding to external stimuli. So I'm talking about them as if they're thinking and doing things by just looking at things, but they're just responding to random events, or these seemingly random events, which makes this really, really neat, I think. Anyway, so that's the difference between MHC1 and MHC2 complexes, and this is why these ones are APCs. They will take in something from the outside, phagocytize it, break it down, and then present whatever it is on its surface to interact with a lymphocyte, to potentially activate it, to potentially start B cell to go in and become plasma cell to churn out antibodies. Question out there?
So I guess how do these MHC2s bind to all these foreign substances?
Okay, well that's a good question. I think your textbook actually goes into a little bit of detail. But basically, these thigolysosomes will have, on the interior, they're actually lined by these MHC1 complexes. So when this stuff gets broken down, it just automatically attaches to this. And once this is migrated out here to the outside to dump the contents, the MHC2s are automatically right there on the surface containing the actual fragments.
Sorry, it was like on one alongside, it must be very non-specific then. Like,
They are somewhat non-specific. So the interaction with the actual antigen is not very specific. But the molecules themselves are very specific in terms of their structure. Okay? So that your own cell surface receptors of lymphocytes, CD4 or CD8 receptors, can actually interact with these molecules and be able to detect them as self as well. So again, we don't have the time to go into all this fascinating stuff in this class because I have a lot of slides to get through. So let's just move along. But again, I encourage you guys to take a look at the textbook and read through a little bit more of that stuff on your own. Okay, so again, a lot of cells that are phagocytic are going to belong to this particular category. Okay? Again, neutrophils would not because they don't really present anything. They don't persist long enough to present anything on their surface. So not every phagocytic cell would be part of this. Okay? But things like, for example, members of the mononuclear phagocyte system, like macrophages, would be also belonging to this category of antigen-presenting cells. There are Kupffer cells, which are basically specialized macrophages, Langerhans cells, as well as cells that are called dendritic cells, which basically look like round cell with lots of cytoplasmic projections going in different directions. Those are highly phagocytic. Some of them circulate in your bloodstream. Some of them are found within your tissues. Then there's a special type of dendritic cell called a follicular dendritic cell that we will be talking about in association with, especially in lymph nodes, that are technically not belonging to that category. So they do present antigens on their surface, but they do it in a different way than using an MHC2 complex, and so they technically do not belong to this APC category. So keep that in mind that, again, not every single cell that presents antigens on its surface is actually classified as an antigen-presenting cell. And we'll talk a little bit about epithelial reticular cells in the thymus. And again, these are antigen-presenting cells, but they are not part of this mononuclear phagocyte system. So it gets a little complicated, I guess. Don't really think about it too closely. So let's talk about the different types of tissues and organs. So the first one on the list is just basically a tissue, which is just called diffuse lymphatic tissue. This is, in many cases, the site of initial response. Right? This tissue tends to be found just underneath the epithelium in a variety of different places. So we have things like gut-associated lymphatic tissue or bronchus-associated lymphatic tissue, which basically are found just underneath the epithelium and lining your digestive tract and lining your respiratory tract. Again, these are parts of your body that are in contact with the outside world. And again, parts of your body that tend to have an epithelium that is not stratified squamous in many cases. And so, if that epithelium becomes thinner, it's easier for pathogens to cross it. Which means that what you want to have underneath that epithelium is a fairly good defensive system. And that's what that diffuse lymphatic system is there for. So, I'm going to show you slides of the digestive tract, specifically the small intestines. And then what we have is a simple columnar epithelium. Again, you just have to pass across one layer of cells. If you can manage to break through some of the junctional complexes in between those cells, you don't even have to deal with the cell itself. You don't have to fool the cell into allowing you entry. You just have to pass in between them. Okay? So it's just a single layer of cells, and then you're in. Okay? And so again, you want to make sure that you have something there on the other side of that epithelium to protect you from any of those pathogens that might be able to actually manage to get across. And so that's what this mucosal associated lymphatic tissue. It's called mucosa because the first layer underneath the epithelium in these organs is referred to as the mucosal layer. Okay? So that's a mucosa associated lymphatic tissue, or MALT. And again, depending on where it is, has a more specific name to it. Okay? And so what we tend to find there is usually that mucosa is a fairly loose connective tissue. So if you recall your lecture on connective tissues, what we have there is a fairly loose aggregation of collagen and some elastin, and we have fibroblasts, some very small blood vessels. Okay? And it's a fairly open meshwork. Okay? And so within this meshwork is where we tend to find a lot of these plasma cells and lymphocytes and macrophages, eosinophils, all these different types of cells that are going to be there monitoring that area for the possibility that there might be a pathogen crossing that epithelium. So if we take a look at this slide here, actually the next two slides, what we actually have is a villus. So I'm just going to draw kind of a zoom out of what we're looking at. So you've got a villus. So this is a villus. It's basically a specialization in your small intestine that's used to increase surface area of that epithelium to allow for easier, more efficient absorption. Now, on the surface of that, we have a simple columnar epithelium. So you'll notice the nuclei tend to be in nice, neat rows. On the surface of that epithelium, you will quite frequently see microvilli or a brush border. And again, because it's light microscopy, we don't usually see a very easily visible set of individual microvilli or individual projections. What we do see is this kind of a thickening right at the surface. Okay? It's kind of a fuzzy sort of edge right at the surface, which is our brush border. And again, that brush border is more clearly visible if you can see it right next to a goblet cell. Within that epithelium, where basically the goblet cell would not have a brush border, and so you could see that there's a difference between the surface of that cell and the surfaces of the cells surrounding it. Now, underneath that epithelium is just your typical loose connective tissue. So what we have is loose connective tissue underneath that. There will be some blood vessels. So quite often what we'll see are fenestrated capillaries, very closely associated with that epithelium just underneath the basement membrane. They are positioned in the perfect place to just take up whatever these cells are transporting across the epithelium. So you're going to have a lot of capillaries. Right down the middle, quite often there's another vessel called a lacteal, which is in fact a lymphatic vessel. So it's a blind-ended lymphatic capillary that connects some of the larger components that are coming through. So for example, things like some of the larger lipids will not be able to enter into the capillaries. Instead, they will travel into the lacteals, be collected by them, taken off, and eventually will be able to enter the venous system later on down the line. Now, lymphatic vessels are vessels that you won't see any red blood cells in. So you're likely to see red blood cells guarding the insides of the capillaries, but You're not going to see any red blood cells in lacteals. The only type of cells you might see in a lymphatic vessel are white blood cells, specifically lymphocytes. So if you're seeing a vessel that contains a lot of red blood cells going in the middle, it's not a lacteal. It's likely to be a Venule. So don't confuse the two. Okay? Lymphatic vessels do not have red blood cells in their lumen. So it is within this tissue here that you will tend to see a lot of lymphocytes. Why am I drawing these nuclei so close together? What can you tell me about the sizes of lymphocytes? Most lymphocytes are what size? I'm not looking for specific dimensions in this case. Small, medium, or large? Small. How do you define a small lymphocyte? The cytoplasm is very minimal. You have a tiny little bit of cytoplasm around the nucleus. So if you have very little cytoplasm, can you pack a lot of lymphocytes close together? A lot of nuclei close together? Yeah. That's what you will tend to see within lymphatic tissues, is you will see a lot of nuclei very close to one another. It will look very granular because the nuclei are so closely packed. Only when you have the lymphocyte becoming activated does it actually accumulate cytoplasm. And if that lymphocyte accumulates cytoplasm, its nucleus will become more separated from the neighboring cells. This will become important very shortly. Okay, so let's get away from the drawing and actually take a look at the next couple of slides. So the first one is showing you this region right here. The next one is going to show you this region right here. Okay, so basically there are two pictures that are side by side of the same villus. Okay, so here's the base of the villus right here. Okay. This is the epithelium. You can see a couple of goblet cells within this epithelium. Again, the nuclei don't look very well organized in this case. Don't worry, it will become easier when we look at the tip. So this is the lumen on either side of that villus. Okay. This is where we have that loose connective tissue. You'll notice there's lots of these granular cells throughout this tissue. These are not fibroblasts. These are lymphocytes. There are some plasma cells in there. There are some monocytes in there, some eosinophils. Okay? If you look carefully, you can actually start to make some of them out. So for example, there's a huge cell right here that has a very large kidney bean-shaped nucleus and a little bit of basophilic cytoplasm. A monocyte. Monocytes will differentiate eventually into macrophage. Okay? A lot of these smaller nuclei you're seeing here are lymphocytes. Again, they're very closely packed together because there's very little cytoplasm. Again, once in a while we'll see a little bit of collagen passing through this. These are very loose connective tissue, so the collagen bundles are very thin and they're kind of loosely arranged. Okay? So they're not very thick, they're not very dense, they're not very regularly arranged. This here that you're seeing is actually a little bit of smooth muscle. You can see that the nuclei here are much more similar to what you would expect to see. The smooth muscle fiber, they're elongated, fairly pale staining, and central within the actual muscle fiber itself. Okay? A little bit of strip of smooth muscle going around in the middle of a villus actually helps to control its shape, so that you can actually pull back a little bit and contract that villus, or allow it to expand and be a little longer and stick out into the lumen a little bit more. Okay, so we can actually control a little bit of the actual shape and length of the villus by having a little bit of smooth muscle here as well. There is a blood vessel passing through. This is the longitudinal section of blood vessel right here. You can see red blood cells passing through. Would you say this is a capillary or a venule? How thick do you think this blood vessel is? What's the diameter of that vessel? Is it one red blood cell across, 2, 1.5? It's maybe 1.5 to 2 red blood cells across. So it's not very thick. It's maybe 15 micrometers in diameter. Probably a bit thicker than a typical capillary, so probably a post-capillary venule. You have another blood vessel just underneath the epithelium here, longitudinal section. And here, the thickness of the blood vessel, the diameter of the blood vessel, is roughly about the diameter of a blood cell. So it's likely to be a capillary cross-section. Again, because this is a villus, because there's a lot of absorption, a lot of transport of things into the vascular system, this is likely to be a fenestrated capillary. Look at the tip. Okay, so that's the rest of this. If you look up here, you can see very clearly a nice neat row of nuclei with simple colon epithelium. If you look carefully at the surface, you can kind of see that, yeah, probably it does have brush border on the surface. Okay. Again, near the actual basement membrane, we have some capillaries. Going down the middle, this is a fairly thick, fairly high diameter blood vessel, likely to be a venule. Okay, so this is not a lacteal, it has red blood cells within it, so you know it's not a lymphatic vessel. And then all of these nuclei down here belong to a lot of the lymphocytes, a lot of the plasma cells, eosinophils, etc., that are making up part of this diffuse lymphatic tissue. The thing about diffuse lymphatic tissue is it doesn't really have much organization. It's just there underneath the epithelium in case anything passes across. In fact, some of the lymphocytes that started out here have actually migrated down here in between the epithelial cells. So in that intercellular space in between epithelial cells, where you have those lateral infoldings that we talked about at the end of the course, some of the lymphocytes have actually migrated across the basement membrane and lodge themselves right there to be as close as possible to that lumen, so that anything that manages to pass across, you've got some lymphocytes right there to greet them, to say, hi, you don't belong here. Okay? Well, they won't actually say that. Okay. This is a low power micrograph of the small intestine. This time it is the jejunum. No, wait, that's not. It's the duodenum. You don't know why I said that. That's okay. You can actually tell which part of the small intestine you're looking at by looking at a slide. I won't hold you responsible for that, because that's stuff that we would have covered in C22, which unfortunately is no longer offered. So you will never learn, I guess, unless you come to see me during office hours, in which case I'll be happy to explain to you all these things. So again, we've got the lumen. We have the epithelium. You can see a nice neat row of nuclei telling you exactly where the epithelium is. Okay, so we've got the epithelium. Remember, these villi tend to have different shapes, and so depending on how you slice the tissue, you might see bits and pieces kind of just sticking out of nowhere, seemingly. Okay? Again, we don't have a nice regular tube. Basically, you've got a lot of projections going to the interior at different angles. Depending on how you slice it, you're going to get some slices of villi that just don't seem to have a base anywhere. Okay? But underneath that, you see all this kind of a very diffuse sort of aggregation of, fairly disorganized aggregation of these dark staining nuclei. Okay? All of this here is diffuse lymphatic tissue until you get to this point here. This is still part of the diffuse lymphatic tissue, but it's a little bit more organized. That is our lymphatic nodule. Okay, so here what we have is, there's a bit of structure to this. So there's a lot of reticular fibers in the area to kind of let these cells kind of sit on that area. But there's a bit of a limit here. Now there is no specific capsule, there isn't a connective tissue that says this is the boundary of this structure. But there seems to be a limitation to the organization here. So there's a limit to how far these things will spread out past that central region. And so you have this very round, very spherical sort of structure. And that's a lymphatic nodule. So it's a bit more organized than typical diffuse lymphatic tissue. And again, these nodules occur in a variety of places, scattered fairly randomly throughout your body. There is one place in the ileum of the small intestine where they actually occur fairly commonly in a very specific way, a very long chain of lymphatic nodules. And that's where they are called Peyer's patches. So Peyer's patch is basically one of these chains, long chains of lymphatic nodules, one next to the other, next to the other, next to the other. And so you might have 20 to 40 of these lymphatic nodules going down the length of the small intestine in the ileum, just underneath the epithelium. So that's a more specific type of arrangement, and so it's actually been given its own name, which is Peyer's patch. Which is mentioned right here. You were wondering how to spell it. Okay. So lymphatic nodules basically are localized clusters of lymphocytes. Again, there is no specific capsule around them, okay, so it's not an organ, but it does seem to have very definite boundaries when you look at it under the microscope. Okay, so it is well defined. It is often associated or very close to this more disorganized, diffuse lymphatic tissue. And it's found very commonly in tonsils. If you look at the pictures of the tonsils that we have in a textbook, you will recognize them immediately. Very, very tightly packed little, relatively circular structures, very closely packed together. And again, small intestine is another place to look for them. That's where we call it Peyer's patches. And again, appendix is another place where you're likely to find a lot of these lymphatic nodules. Now, they can be subdivided into primary and secondary nodules, depending on their level of activation. So, a lymphatic nodule that has never seen an antigen is likely to look like this. Basically, a whole bunch of small lymphocytes closely packed together. What do you think a secondary nodule is likely to look like? One that has become activated, one that has seen a pathogen or an antigen. That nodule is likely to contain some large lymphocytes within it. And that's what we tend to see. When we look at a secondary nodule, we have an outer layer, a mantle, an outer rim of small lymphocytes that are tightly packed and pretty densely, pretty intensely staining. And then a central region called the germinal center, which is where we have proliferation of lymphocytes and an increase in the size of the lymphocytes. So that's where we can have the large lymphocytes. And again, if you think about this, large lymphocyte means lots of cytoplasm around it, which means the nuclei of those lymphocytes cannot be as tightly packed together. If you look at that region under the microscope, a lot more light will be getting through to your eyes because there's fewer nuclei in the way, fewer intensely stained things in the way. And so it will look more pale staining. And so what we tend to find is that the germinal center has a very light, very pale stain because of the fact that you have large lymphocytes there in that area as opposed to small lymphocytes, which are much more tightly packed, and so a lot more light is being blocked before it gets to your eye. And so again, here is an example of a primary nodule or a couple of primary nodules. These are in fact Peyer's patches. And then I don't have a secondary nodule to show you right now. I think I have one a little further down the line. Let's move on to the lymph node. A larger structure before we move on. Yes?
I just had a quick question.
Yep.
You said that these lymphatic nodules are from mostly the appendix and tonsils.
No, those are where you're likely to see a lot of them very close together. They're very obvious there. But they are scattered throughout your body. So
I was
it's a very
wondering,
random sort of arrangement.
is there like a specific reason, like because I know the appendix and tonsils can be removed like, due to inflammation and stuff like that. So is there an association with the number of nodules that are there?
So is there an association with the number of nodules and the removal of the structure? About the question, you have an answer.
Well, no, I was just wondering more along the lines of his question, like,
Okay.
if tonsils are removed because they're inflamed, so like these nodules, they can't switch between primary and secondary, like if an antigen is gone, it won't revert back to the primary.
Okay, so along those lines, so let's talk about tonsils. If you have tonsils, and imagine this, but hundreds of nodules very closely packed together. That's basically tonsils. Okay? And they're basically lining the back of your oral cavity, okay? Right where any kind of pathogen might be entering your body from the outside. Right? So you definitely want to have tonsils there. They will be monitoring the system. So if we're going from a primary nodule to a secondary nodule, do you think the nodule itself is likely to increase in size? Remember, we're having lymphocytes that initially were small lymphocytes, suddenly becoming large lymphocytes, and also proliferating, so more large lymphocytes. Are those nodules going to increase in size? Yeah. Okay. Imagine that going on around your throat in hundreds, if not thousands of nodules. What's likely to happen at some point? going to swell up a lot. You won't be able to breathe after a while. So this might be why tonsils would be removed in some individuals. I don't think necessarily it's something that we want to be doing, okay? Because again, it's, I think, a fairly important part of our monitoring system of whatever's coming into our body. But in some cases it has to be done because there's the danger that the airway might become blocked and the person can't breathe. And breathing's probably a little bit more important to you than being able to catch every single pathogen right there at the very entry point. Okay? In terms of appendix, you don't remove the appendix unless there's inflammation there. Okay? And if it's in danger of really affecting things, because if that thing bursts, you have inflammatory response going on. Well, you have things spilling out in places where they're not supposed to go. Okay? Normally, under normal conditions, again, you wouldn't remove your appendix. Your appendix actually is not some vestigial organ, as you may have been taught in high school. It actually does have a function. It does play a role in the body. Right? But sometimes it is important to basically protect you from this thing bursting and spilling its contents all over the inside of your abdominal cavity, where you might need to have it removed. Okay? Is there a relation between that and the number of lymphatic nodules there? Maybe. Okay? Again, especially with the tonsils, you got a lot of lymphatic nodules there. They all start to swell up. Well, that's going to be a problem. But again, that might probably be more of an issue there with just your sensitivity to certain antigens, not so much the fact that you have lots of nodules. We have developed these nodules over millions of years for a reason. Okay? So they're not there to hurt you. They are there to help you. And so again, removing these things is not really something that we should be doing necessarily unless it's absolutely necessary. Okay? Okay, one more question.
I was just wondering why the center of the nodules are more active than the outer rim.
Why is the center more active than the outer rim?
Being closer to the lumen, that's more active.
It's interesting. It's a good question. I think part of it is because the cells that actually bring the antigen into the region look to migrate towards the center of the actual nodule. Right? So these antigen-presenting cells, these macrophages and these dendritic cells, would then make their way into the center of the nodule and activate the cells there. Okay, let's move along because there's a lot of slides to get through. Not a lot of time. So the lymph node. So our first lymphatic organ, this is the smallest of the lymphatic organs. It's about a centimeter in length, maybe, if it's large. So they're not terribly big. Actually, not a centimeter, sorry. Of course, I would decide not to write that part down for myself. They are not very large, but they are visible with the naked eye. If you do see them in a dissection, for example. So those of you who took anatomy, you've probably seen a whole bunch of lymphatic nodules. Sorry, lymphatic nodes, not nodules. So it is a lymphatic organ because it does have a capsule surrounding it. So it does have a definite limit to it. Its function is basically to filter lymph. Okay, so again, look back at your picture that we drew earlier of the vascular system and the lymphatic vessels. And everything that is taken up by the lymphatic vessels will be drained into a lymph node along the way, at least once. Okay? So, before that content of the lymph enters your vascular system again, it has been cleaned up. Any pathogens that have been in there have filtered through 1 or 2 or 3 lymph nodes before they are dumped into the vascular system. So it does a lot to basically filter things out. It's a filter. So interaction of antigens with lymphocytes to initiate immune response is one of the primary reasons for that lymph node to be there. Now, if you think about it this way, one B cell, B lymphocyte, is equal to one type of antibody. Okay. That antibody only detects one thing. Okay. So one plasma cell equals one antibody. Okay. It's only sensitive to one thing. One T lymphocyte is really only designed to detect one thing. So it's going to have receptors on its surface, but it's not going to be able to detect every single one of the millions and millions and millions of different antigens that we come into contact with. Okay? So it can only detect one or two things. Okay? So if you want to initiate an immune response to an antigen, is it better to bring the cells, all of your B or T lymphocytes, to that antigen? Or is it better to bring the antigen to a place where you have lots of B and T cells? That second option is what the lymph node is all about. Okay, so what we basically have in a lymph node is a collection, a huge collection of B and T lymphocytes. Now, these cells are able to respond to thousands, if not millions, of different antigens in that one lymph node. So when you have fluid draining from an area of inflammation, it's going to have a whole bunch of different antigens within that. Those antigens will go through and enter that lymph node. They will filter past a whole bunch of lymphocytes. Most of them probably will not recognize that antigen. They will not say, they will basically, I don't know what this is. I can't touch it. I don't know what to do with this. There might be one or two that might say, oh, I know this. This doesn't belong. I can attach to this. Once they become activated, they can start to proliferate. They can start to make more copies of themselves. They can start churning out antibodies if it's a B lymphocyte, okay, specifically against that antigen. Once they become activated by that antigen, they can say, okay, now I become active. I make more copies of myself so that I can respond to this threat in a better way, a more efficient way. Okay? And so again, what lymph nodes allow you to do is filter a whole bunch of antigens past a small cluster of cells that contain many, many different types of B and T cells. So, activation and proliferation of B lymphocytes will be one of the things that's going to happen there. Again, same thing for T lymphocytes. And again, if you're activating B lymphocytes, you're going to make plasma cells that produce antibodies. And, of course, macrophages are going to be present there as well to remove whatever's passing through. So, the structure itself. The lymph node is encapsulated. It has what is known as a discontinuous capsule. Why is it discontinuous? Well, let's draw. You can stick with this slide. I'll do some drawing. So, let's say this is our lymph node. What I'm drawing right now is the capsule of that lymph node. The capsule is composed of connective tissue. Remember, this is a schematic diagram. It's not going to look exactly like this on the slide. It's just meant to give you some idea of the structure itself. So this is the capsule. It is discontinuous because it has a lot of lymphatic vessels coming in from all directions and draining into it. Now, lymphatic vessels do have valves, just like veins. Lymphatic vessels are, in fact, very similar to veins in terms of their histology, although they are somewhat thinner in terms of their walls, even than veins. And so what we tend to see is that we do, in fact, have valves coming in. In fact, lymphatic vessels actually have more valves than veins. So they're more frequent in lymphatic vessels than they are in veins. I want you guys to notice one thing. This one right here, I'm going to draw the valves in a different direction. Because most of these vessels lead into the lymph node, while one of them leaves the lymph node itself, so leads out. Okay, so this is an afferent. So we've got multiple afferent vessels. Many. have one efferent. So that means they have many different sources of fluid from different tissues coming in to this one lymph node, all draining into one lymph node. And then all that fluid gets collected here, tested for the presence of antigens that do not belong, and then the rest of it is about to flow out once it's been filtered. Now, some of the things I've drawn here. This right here is the trabecula. Here's one. So the trabeculae. Seen this term before when we talked about bone, okay? But it's not specific to bone. Basically, a trabecula is a solid mass of material. That's what we're seeing here. This connective tissue from the capsule invaginates into the interior to give the lymph node some structure. Basically, it's an open, kind of an empty shell. That shell needs a little bit of structure so it doesn't collapse in on itself. And so there are these trabeculae there just to hold it open. There's a bit of stuff there to hold it open, kind of a network, scaffolding which holds this thing open and available for cells. Now, underneath that capsule, we tend to find an initial region packed with lymphocytes. And the lymphocytes tend to be organized into nice round structures called nodules. So we actually will once again see lymphatic nodules within a lymph node. Generally, students are releasing lymph nodes again. So, lymphatic nodules found underneath the capsule in a region we call the cortex. So this region right here, where we see nodules, it's called the cortex. Now, underneath the nodules, there isn't necessarily an empty space. What we have instead is kind of a more diffuse aggregation of lymphocytes. Doesn't really have any kind of shape to it. They're fairly tightly packed still. And so that region is called the paracortex. And then the interior, draw them in color. The interior is the medulla. So the trabeculae is basically the solid material that is used to hold the actual nodule open. Nodule is basically just a bag of lymphocytes, essentially. Okay? That bag of lymphocytes receives a lot of fluid coming in from different tissues and then releases fluid out again. Okay? But it needs to have a little bit of structure to it. So basically, you can think of these trabeculae as things that will kind of just hold it open. Okay? Now, within the actual nodule itself, in the cortex and paracortex, etc., there's going to be a network of reticular fibers. And you know what those look like from your box or from our discussions of connective tissue. And these are argiophilic fibers. And again, they're acting as a scaffolding on which the cells will sit. Okay? So we've got reticular fibers. In fact, if you go back to your slide of reticular tissue from your connective tissue lab, you'll actually notice that you're looking at a lymph node. Okay? And so if this is reticular tissue, that means we have reticular cells that produce it. So the cells in there, one of the cells that's going to be in there is going to be the specialized fibroblast called a reticular cell. Okay. And so again, it is on that network of reticular tissue, of that reticular fiber network, that you will have these lymphocytes sitting, and then what you have is this flow of lymph kind of percolating through this, filtering through this, and passing by all of these lymphocytes. Now, one of the things about what's going on in that cortex is that in the cortex what we have, let me just go back to our slides. Okay. In the cortex what we have are lymphatic nodules which contain B lymphocytes and plasma cells, as well as these follicular dendritic cells that I mentioned to you a little while back. Okay. These are cells that do present antigens on their surface, but they're not presenting them on an MHC2 complex, so they're not technically part of the APC category. Okay. And so those follicular dendritic cells are basically these star-shaped cells. You've got nucleus, you've got a cell body around the nucleus, and you've got lots of these cytoplasmic projections, very long projections going off in different directions within a lymphatic nodule, and they will be collecting the antigens that might be suspended within that lymph. They will be collecting it and then presenting it along their surface to all of the B cells that they're in close contact with. So that those B cells don't have to be right near the edge of that lymph nodules. They can be in the middle. That follicular dendritic cell is going to bring the antigens to it. So let's go back. So again, we have a reticular tissue network produced by reticular cells. We have afferent lymphatic vessels coming in, then we have this efferent lymphatic vessel leaving at this region called the hilus. So this region right here is referred to as the hilus. And again, it's not turned out specific to lymphatic nodes. There's a hilus in your spleen, there's a hilus in your liver. Basically, it's kind of an indentation in a particular organ through which you're going to have the vascular system coming in and leaving. So one of the things that happens here is that you will also have an artery coming in. So we have an artery, passes through, opens up into a vascular network, a capillary network up here in the cortex, and then perhaps drains into venules, and we've got a vein leaving. So we do have a vascular system feeding all of this as well. So we do have an artery coming in, opening up into a capillary network, especially around the cortical region, and then draining into venule system, and then you've got venules coming back down and leaving as a vein from the actual lymph node itself. Okay, so again, you've got one artery, one vein, and one efferent lymphatic vessel, basically penetrating the structure at this particular point. We'll get back to the vascular system shortly, by the way. Okay, so again, we have the division of the cortex, pericortex, and medulla, and if you look at this slide here, we're looking at a lymph node, kind of a longitudinal section here. And basically on the outside what we have is the capsule. Again, my drawing was much more schematic than it might seem. The capsule here is not very clearly visible. We don't have a lot of staining here, unfortunately. What we have underneath the capsule is you can see some of these nodules, these round structures. Notice that the central regions of these nodules are very pale. These are secondary nodules. This is clearly a lymph node which has already met an antigen. Okay. So a lymph node that has already been filtered through and has had to make antibodies against something. Okay. So these guys here are all secondary nodules. Okay. So what we find in the cortex is mostly B lymphocytes and these follicular dendritic cells. This is where you would have antibodies being produced. This is where you would have B lymphocytes becoming activated to become plasma cells. Again, those follicular dendritic cells will tend to sit somewhere near the middle of this, but have their processes extending out in all different directions and collecting antigens as they are percolating through towards the center of this structure. Now, once the lymph passes through the cortex, it will enter the paracortex. The paracortex doesn't have nodules, it just has lymphocytes and macrophages. And in this case, the lymphocytes are T lymphocytes. So you've got B cells in the cortex and the nodules, and then T cells outside the nodules in the paracortex. This structure right here, you can see an indentation here. This is the hilus of this particular lymph node. Remember, lymph nodes are three-dimensional structures. When you're looking at them under the microscope, you won't always be able to see the hilus. Don't be surprised. They're a fairly large three-dimensional structure. The hilus is just a small point at one point on that particular organ. So don't expect to see it every single time. Not every slice will contain a hilus. This region right here that kind of looks like it's more pale staining and seems to have lines passing through it, this is the medulla. It looks a little odd. I think I just talked about these. So here's just a close-up of another lymph node. Here you can actually see the capsule very clearly. And you can see some of these trabeculae invaginating into the interior to form that kind of a structure, that scaffolding, which holds the structure open. Now again, all of these we have here are lymphatic nodules in the cortex. And then we go deeper in, it becomes a little bit more diffuse. This is the paracortex, and then this more light staining region here would be the medulla. Okay. The medulla does have an organization to it. There are regions that are pale and regions that are darker. The darker regions are known as the medullary cords. And the spaces that you're seeing here, the pale staining regions, are medullary sinuses. Medullary sinuses are basically the tunnels through which the lymph will drain. This is the open spaces. The cords, these darker staining regions, are basically the walls of those tunnels. You can think of it as a subway tunnel. The wall itself is more dense, it's densely packed, it's more cellular. Whereas the tunnel itself is empty, at least most of the time. And so what you have is your lymph flowing through that. Okay, so again, medulla has medullary cords and sinuses. And basically the cords are composed of, again, reticular cells, which produce the reticular network on which everything else sits. And then you have also B cells, macrophages, especially plasma cells, and especially the macrophages. How many of you have seen Indiana Jones movies? The good ones, not the most recent one. That was just the good ones where you have him running through and there's spider webs all over the place. Those are the ones I'm talking about. Okay? You can think of the medullary sinus as one of those tunnels or caves that he's running through with all those spider webs. Okay? The spider webs are produced by macrophages. Macrophages sit within the cords of the medullary sinuses, but they will send out cytoplasmic projections into the sinuses. And so what you have is this meshwork of these projections going back and forth across those sinuses. So if you can think of this, you can have macrophage here, and they're both going to be sending off projections across that open space. And so that means that when the lymph is passing through, those cytoplasmic projections will be able to catch whatever is suspended within that lymph and filter it out. So if it hasn't been filtered out in the cortex or the paracortex, it will be filtered out here as well. So you can think of one of those sinusoids, the medullary sinuses, as being very much like one of those caves through which Indiana Jones might have to run, where you have all these spider webs going across, and it makes it a little difficult to pass, and it's easy to get caught up in these things. That's kind of what these sinuses are like. Yeah.
It's so since it's within the reticular network and it passes projections through the sinuses.
Yeah. So what you have is a reticular mesh, reticular tissue. Within that reticular tissue, you're going to have regions that are more densely packed with cells, and those will be the cords, the medullary cords. Then you have more open spaces, which would be where you don't have as many cells. The reason you see nuclei here is because there will be some cells that are suspended within the lymph. For example, you might have B cells that proliferated out here, but now need to get out and be distributed throughout the body. That will then be able to enter this lymphatic flow and then be taken into the venous system and be recirculated throughout the whole body. Same goes for T lymphocytes as well. So here's a slide of silver-stained lymph node. This is taken from the reticular tissue slide. And again, if you look closely, there will be regions that you can see are far more cellular, are much more densely packed, and regions that are a little bit more pale staining because there's less stuff there. And again, this will be one of these medullary sinuses. This will be one of the cords. This will be one of the cords over here. Okay, before we move on, one last thing. How does the lymph flow? Has anybody thought about that? I mean, we know blood flows because there's a pump pushing it forward. And then there are muscles milking the veins, pushing the blood back towards the heart. How does lymph flow? Sorry? Well, it takes up the interstitial fluid, but why does the interstitial fluid flow towards a lymph node? Diffusion. Well, okay. So here's the answer. Back to this vascular system here. So what we have in here is something that I haven't told you about, called an HEV, or high endothelial venule. So once in a while, I tell you guys that I am really simplifying things, and there are exceptions to what I tell you. This is one of those exceptions. I have told you in the past that the endothelium is a simple squamous epithelium. And for the most part, that's true. Here it isn't. Okay? So a high endothelial venule is called a high endothelial venule because it has a high endothelium. It actually has an endothelium that looks a bit more columnar than squamous. Now, why do cells normally need more cytoplasm? Because they're more active. They're doing something. They need that cytoplasm. They need some organelles to do some sort of function, perform some sort of function. Okay? And that's the case here. So right around the paracortex, right around this region right here, in the lymph node, is where we tend to find high endothelial venules. Now, they have two functions. First of all, they allow for fairly easy transmigration of lymphocytes across that endothelium. So, in the lymph node, the T lymphocytes have to get there somehow. They got there through the high endothelial venules. They enter the paracortex, they leave the thymus, they get into a lymph node, and they exit the vascular system through the high endothelial venule. They are able to pass across the endothelium fairly easily in between the endothelial cells. Okay? So, easy passage for T cells. But the other part, the one that we started off with, is the flow of fluid through the lymph node. If this is a high endothelial venule, this is the paracortex out here. Okay. Then what you have is you have the lymph fluid passing through the lymph node. A lot of that fluid is going to be water. That water is going to be actively pumped into the lumen of this venule. So water that's in the lymph is going to get pumped into the lumen. Now this obviously is going to require energy, and so that's why these cells are a little bit taller than a typical endothelial cell. Okay, so you're going to have very active pumping of fluid out of the lymph node and into the vascular system. What's that going to do?
Drop the pressure.
Sorry?
It would drop the pressure.
Drop the pressure.
It would bring in more liquid.
It would definitely bring in more liquid. Why? Because of the thing you learned in first year chemistry. Adhesion of water molecules. They're interacting with one another. There's hydrogen bonding there. And it pulls water along as it's being removed. Right? The same reason that trees can get water all the way up to the very top. Because it evaporates at the leaves, and so more water gets drawn into the tree. Same idea here. If you remove water out of the system here, more water will get drawn back into that lymph node. And that's how you have the removal of water from the tissues, by basically creating this solvent drag. So solvent drag is how lymph gets pumped. By basically removing water from the lymph node, more water is going to be pulled into the lymph node to basically follow that. Now, please don't ask your TAs to find you a high endothelial venule. Look for them yourselves. I don't want your TAs spending an hour trying to find you a high endothelial venule and say, this is a technical example. Okay? So try to find one. If you do, tell everybody else, and then you won't be able to use your microscope for a while while everybody else is looking. Okay? All right. So let's move on to the thymus. Thymus is the second largest lymphatic organ. It is a bilobe lymphatic organ. It's just anterior to the heart. It's a transient structure, so it does change over your lifetime. So, for example, in me, my thymus is probably fairly shrunken and fairly inactive at this point. In your case, it's just getting there. You're almost done using your thymus. Okay? Now, your thymus does atrophy and become replaced by adipose tissue. This is called involution. But it can be re-stimulated in some extreme cases. So it can return back to its function if your body absolutely requires it. But it is very, very important in development. So if you remove it too early during development, in childhood for example, then you could end up with an individual who is immunocompromised. So it is very, very important because it's responsible for educating your T lymphocytes. Your T cells start out as thymocytes. These cells in the thymus that don't know anything. Thymus is basically a school for thymocytes that tells them this is the difference between self and non-self. This is how you recognize an antigen. Okay? Basically teaches them to look for MHC2 and MHC1 molecules. Teaches them to look for what's inside of those molecules. And teaches them how to recognize whether it's self or non-self. All of that is happening in the thymus. If you don't have the thymus, you can't teach your T cells that, and so your T cells are not going to function properly. Okay, so initial slide of production and maturation of T lymphocytes. So you're going to have a progenitor, a stem cell of the T cell that will initially develop in the bone marrow and then migrate into the thymus, and that's where it's going to proliferate and start to develop into the T lymphocyte that's going to be released in the end. So you're going to have this development process occurring here, which is what we'll be talking about next. It does have some endocrine function. It produces two hormones, thymosin and thymopoietin, which are basically going to be used within the thymus itself to help steer the development of the lymphocytes themselves. Now, the structure itself is encapsulated It has a dense irregular connective tissue capsule. So anytime we have a capsule, basically it's a dense irregular connective tissue capsule. Okay. Again, the capsule does invaginate into the interior of the organ to produce septa. It's kind of like trabeculae, but they usually refer to as septa. Basically, you can think of the thymus as being this kind of almost a heart-shaped structure. So this is the capsule. Again, that capsule invaginates into the interior to break it up into lobules. And we'll talk about why in a moment. Okay. So let's draw this thing first. So you've got capsule, you have septum, or septa plural, which subdivide the thymus into lobules. Lobules have a cortex and a medulla. They will kind of look like nodules, but the thing to remember about the thymic lobules is that they are all interconnected. So you don't actually, even though it might look like it on the slide, you don't actually have individual round structures that have a germinal center. What you have instead is this dark staining material around the outside, so the cortex, and the lighter staining in the middle, lighter staining region in the middle, which is the medulla. And of course, that's too light. Now, the reason why you have this subdivision for these lobules is that the majority of the lymphocytes, the immature T lymphocytes or thymocytes, are at the periphery in the cortex. Okay? And about 98% of them will never leave the cortex. In fact, they will be killed. They will die. Only about 2% of the lymphocytes in the cortex actually graduate. So the thymus is a very, very strict school. Only 2% of students actually manage to get out. Everybody else fails. It's not a very good rate. Very, very tough. Must be a very prestigious institution, I guess. So only about 2% actually ever make it out into the medulla. Okay. Which means that what you want to have is a large amount of space for the cortex. You don't need a lot of space for the medulla, which is why this organization occurs. Because having these septa increases that area that's available for having a cortex, okay, and allows for a much smaller medullary region. Yeah.
So, like, whatever lymphocytes are like in the cortex, they're not fully educated yet, so they can't actively respond.
They're not going to be responding to anything in your body. You do not want to send those cells off to do anything in your body because they don't know the difference between self and non-self yet. Okay? So the thymocytes that are in the cortex are basically stuck there until you release them. And we'll talk a little bit about how that happens. Okay? All right. So you have a structure that is encapsulated, has septa or trabeculae that break it up into lobules, not nodules. Okay? And basically the lobules contain a support network, but that support network is not composed of fibers. That support network is actually composed of cells. And these cells are called reticuloepithelial cells or epithelioreticular cells, depending on which source you go to. Okay? So we'll go with reticuloepithelial cells because that shows to RECs. So no reticular fibers, no reticular cells. What we have instead are reticuloepithelial cells, which have properties of epithelial cells and properties of reticular fibers, which is probably why the name came along. Now there's many different types. There's 6 different types of epithelial cells. We'll talk about each one of those in sequence. It is a very highly vascularized organ. You will quite often see blood vessels passing through it. It has no afferent lymphatic vessels. This is not a filtration system. This is, again, a primary lymphatic organ. So it's not there to filter anything or to respond to any kind of a pathogen. It's just there to educate lymphocytes and release them into your circulation. Okay, so it's going to be highly vascularized. In that medulla, where you're going to have the mature lymphocytes, the medulla is going to have some high endothelial venules, or HEVs, to allow those lymphocytes easy access to the vascular system so they can actually enter and be distributed throughout the body. And again, divided into cortex and medulla. So this is a low-power micrograph of a fairly young thymus. And so what we're seeing here is a capsule invaginating into the interior to form these lobules. So this is a lobule, this is a lobule. And again, notice that in some cases they kind of look like nodules, like secondary nodules. Again, remember, this medulla here is continuous with this region here, and this region here, and this region here. So all of these are continuous with one another. So they're not in fact spherical structures. What we have is just a lot of these invaginations, these deep invaginations, to allow for more presence of a lot of cortex. So you have lots more room for these immature thymocytes, these immature T lymphocytes. Okay? So again, the darker region here is the cortex. It's darker staining because you have lots of small lymphocytes packed very closely together. Whereas the medulla, much more or much less intensely staining because you have fewer cells there, you have fewer lymphocytes there. Okay, so it's a much less cellular region. And so it picks up less stain because more light passes through to your eye. Here is the thymus of what it might look like in you right now. Okay. So this is what involution looks like. Okay. Same thing, you still have the nodules, but a lot of those that tissue has become replaced. So you can still see some of these septa passing through this tissue, but you can also see there's a lot of adipose tissue replacing that lymphatic tissue. Okay? This is a process that occurs in the thymus. It's also a process that occurs in your bone marrow. So that's how your red marrow becomes yellow marrow. Okay? This is why some of the slides that you saw in your slides of bone, for example, has a lot of fat in the bone marrow. You can see a lot of fat cells in it. Okay? That's because involution is occurring. So the cortex is packed with very tightly packed, very densely packed lymphocytes, or really immature lymphocytes, or thymocytes. And these cells have no idea what they're supposed to be doing. They are fairly powerful. They are quite capable of causing damage, which is why you want to keep them separated from everything else. And so the cortical region is really all about isolating these thymocytes from everything else. So let's go to another drawing. So let's kind of zoom in on like a region right here, let's say, and just blow that up a little bit for ourselves. And so what I'm going to do is, I'm going to flip this so that it's going from top down. So here's my capsule, let's say. And I've got a septum going into the interior. Here's another septum. Again, the septa will tend to contain blood vessels within them. And those blood vessels will then eventually enter into the actual tissue itself. Now, again, because the lymphocytes here could basically react to anything, you want to isolate them. Because you do not want them reacting to your connective tissue. You don't want them reacting to your capsule and trying to destroy it. So you're going to isolate them. And so what you're going to have is a layer of cells. I should judge a different color for that so we can see them better. Okay, so we've got a layer of cells, which is which kind of look like epithelial cells, and in fact have characteristics, very reminiscent of epithelial cells, in that they have tight junctions. They have the zonula occludens, very elaborate zonula occludens, which basically means that they don't allow things from the outside to pass in between them. So that's stopped. Okay, so what we have is junctional complexes connecting all of these cells together, and the only way anything passes across into the cortex is by permission from these cells. And these are type 1 RECs. If there's a blood vessel passing through, so there's a blood vessel, let's say, that periodically happens. The outside of the blood vessel will also be covered by a layer of type 1 RECs. Now, don't draw that layer too close to the blood vessel, because in between the blood vessel and these type 1 RECs is going to be a layer of macrophages as well. So you've got macrophages. Get a sharper brown line. So again, type 1 REC here, and let's say here. Okay. Thing in red is the blood vessel. So another cell type that's going to be in there, in between all of these, is going to be a type 2 REC. These more star-shaped sort of cells. Type 2. There's going to be a layer of type 3 RECs down here. At the boundary between the cortex and medulla. Okay, so this here is the cortex, down here is medulla. Oops. You didn't get much room, did you? Okay, so why? Well, again, the type 1 RECs are there to isolate your thymocytes. So anything that could potentially be something that the thymocytes could respond to and potentially damage that you don't want them to be responding to is going to be isolated by these type 1 RECs. Type 2 RECs are the teachers. On their surface, they're going to present good old MHC1 and MHC2. And they're going to present some self-antigens within those. Okay. The job for these lymphocytes here is to be able to recognize and bind to MHC1 and MHC2 and recognize that there's something inside them. It's called double positive selection. Only thymocytes that can recognize both MHC1 and MHC2 will be able to graduate from the cortex. Now again, these cells are going to produce a variety of different proteins, and so those are the proteins that they are going to present on their surface. So they're going to be presenting self-antigens. So we're not too concerned with these thymocytes detecting self and non-self here. Okay? Well, all we want to know is, can this lymphocyte here, potential T lymphocyte here, can it detect and bind to an MHC1 or MHC2 molecule? Because that's the first step you need to have in order to even look for a foreign antigen. If it can do that, then this cell will be able to pass across two layers of cells, the REC3 and REC4, type 4 REC barrier, okay, which is basically the barrier between the cortex and medulla. Okay. Why do we need two layers? Well, you don't want to open up just one layer of cells and allow things to pass across. You're going to have a cell out here. These cells here will kind of go around it, allow it to enter into this space, and then these cells will allow it to enter the medulla. So you've got control over which cells are allowed to go, which ones are not. Okay. The cells that fail this test here, the double positive test, are going to initiate apoptosis. They have about 3 to 4 days to recognize both of these. If they don't accomplish this within that time period, they're going to go into apoptosis. They will die. They will be taken up by macrophages. So there's going to be macrophages here as well. If they do manage to pass the test, they enter into the medulla. The medulla obviously is much less populated because 98% of the cells roughly do not make it out. So 90% of the cells fail this test. Okay. The cells here in the medulla are going to be tested again, this time by dendritic cells and by macrophages. Okay, so we're going to have macrophages and dendritic cells. So it's a dendritic cell. Are you going to be able to see a dendritic cell on the slide? Probably not. You can't see the little processes under the microscope because they're just cytoplasm and plasma membrane. Okay? And macrophage. Okay. So these cells are now going to present MHC2 complexes, and they're going to present self-antigens. These cells are going to produce what is a single negative, so negative selection. So on their surfaces, they're going to have, again, MHC2 complexes, but they will be presenting self-antigens. Okay? That seems a little counterintuitive, but here is the logic. If a lymphocyte attaches to MHC2 complexes, which is supposed to, and becomes activated by a self-antigen, then you do not want that cell there. Okay? So if a cell responds at this stage here, that's a bad thing. Okay? So these cells will be eliminated. Any cell that responds in the medulla to a self-antigen with an MHC2 complex is going to be eliminated. Otherwise, if you don't do this, and these cells enter your vascular system, you're going to have issues with autoimmunity. So you want to remove any cells that would react to a self-antigen. So any cells that do not react, which is why it's a negative selection, are able to enter the vascular system and be distributed throughout the body. Okay, so one last thing on the thymus. I drew this blood vessel here for a reason. Okay. The thymus has something called the blood-thymus barrier. Oh, one last thing also. The Hassel's corpuscle is a characteristic feature of the thymus, okay, or the medulla specifically. In the medulla, what you will have is something that kind of stains fairly eosinophilic. That's going to be filled in It almost looks like a blood vessel, but it seems to have multiple layers of cells, and then maybe something kind of just sticking out. Okay? These are type 6. Type 6 RBCs. There's also type 5, which is going to be used to clean up a little bit in the medulla. Yeah. So type 6 are the ones that produce the Histological corpuscle, which is a relatively large structure. If you see it on a slide, you know you're looking at the thymus. Okay? So this right here is a slide of thymus, relatively high power. Let's turn down the lights a little bit. Okay? And so you can see more intense staining in this region here. That's the cortex. This region here is a bit more pale. That's the medulla. And this structure right here looks like a blood vessel kind of filled in and stuck around it. This is a Histological corpuscle. And those are type 6 RECs. Those are the easiest ones to identify. The other ones are going to be difficult. Right? If you're looking for type 2 RECs, they will again be found in the cortex, and they will have fairly pale nucleus because they're fairly active. The chromatin is very active. Yeah.
So what are type 6 responsible for?
They are responsible for. We don't really know exactly. One of the things that they do seem to produce is those hormones, the thymosin and thymopoietin. That seems to be one of the reasons for having a Histological corpuscle. Although there seem to be other molecules that they produce as well that might be involved in also guiding the development of these lymphocytes. So the blood supply itself has a blood-thymus barrier, which again is there just to prevent any outside molecules from coming in. So again, it has to be separated. So you're going to have an endothelium that is a continuous endothelium, continuous capillaries. It's going to have a continuous basement membrane. The tight junctions on those endothelial cells are going to be very elaborate. You're going to have a layer of macrophages around the outside to phagocytize anything that actually does manage to cross that endothelium. And then on the outside is going to be a layer of type 1 RECs again. So that blood-thymus barrier is very, very important as well. Okay, so we didn't get a chance to talk about the spleen today. So obviously that's not going to be on the quiz, but your TAs will probably cover it. So please make sure you read through the slides before you turn to the lab. And your